Hi everyone, the topic is DNA fingerprinting. DNA fingerprinting was developed by Alec Jeffries in 1985. DNA fingerprinting involves identifying differences in DNA among individuals. This is identification of specific region of DNA called repetitive DNA. When the bulk DNA subjected to density gradient centrifugation, a small portion of DNA will get, that is called a satellite DNA. Though satellite DNA does not code for any protein, it is used as the basic of DNA fingerprinting. One of the most important basic of DNA fingerprinting is RFLP, that means restriction fragment length polymorphism. Let me can see how DNA fingerprinting can be done through the following slides. In satellite DNA, repetition of bases is in tandem. Depending upon length, base composition and numbers of tandemly repetitive units, satellite DNAs have subcategories like microsatellites and mini satellites. Satellite DNA show polymorphism. Polymorphism arises due to mutation. The junk DNA regions are thus made up of length polymorphism, that is RFLP, which show variation in the physical length of the DNA molecule. Next, we can see what is STR and VNTR. Specific loci on the chromosome, the number of tandem repeats varies between individuals. There will be a certain number of repeats for any specific loci on the chromosome. Depending on the size of the repeat regions are classified into two groups. One is short tandem repeats or STRs, contain 2 to 5 base pair repeats. Next one is variable number tandem repeats, VNTR, have repeats of 9 to 80 base pairs. Since a child receives 50% of the DNA from his father and the other 50% from his mother, so the number of VNTR is at a particular area of the DNA of the child will be different, maybe due to insertion, deletion or mutation in the base pairs. As a result, every individual has a distinct composition of VNTRs, and this is the main principle of DNA fingerprinting. A single change in nucleotide may make a few more cleavage site of a given nucleotide or might abolish some existing cleavage site. Thus, if DNA of any individual is digested with a restriction enzyme, fragments pattern will be produced and will be different in cleavage site position. This is the basics of DNA fingerprinting. Next, we can see the methodology of DNA fingerprinting in detail with a schematic diagram with explanation. This figure explains the steps in DNA fingerprinting. The first step in DNA fingerprinting is to isolate the DNA from the sample material to be tested. The sample size for RFLP test must be large enough to get the proper result. Then, once the required size of the sample is available, the DNA is isolated from the sample and is subjected to restriction digestion using restriction enzymes. This is the second step. In the third step, the digested DNA sample is then separated by agarose gel electrophoresis, in which the DNA is separated based on the size. The next step is transfer of separated DNA from gel slab onto the nitrocellulose membrane to hybridize with a labeled prop that is specific for one VNTR region. That means radioactively labeled complementary sequence of VNTR region nucleotide sequence. This technique of transferring and hybridizing DNA onto nitrocellulose membrane is known as southern blotting, a most widely used DNA detection technique by molecular biologists. After the hybridization with the radioactive probe, the X-ray film is developed from the southern plotting, and only the area where the radioactive probe bind will show up on the film. Now these bands, when compared with the other non-sample, will give the final result of the DNA fingerprinting. In short, the 6 steps are given right side of the figure. This slide explains the final result of DNA fingerprinting. The first band shows the DNA obtained from the crime scene. Then there are 3 suspects: suspect 1, suspect 2 and suspect 3. From these suspected persons, the DNA which match with the crime scene DNA is suspect 2. In suspect 2, all the bands are similar to crime scene. Hence, the criminal is suspect 2. Advantages and limitation of DNA fingerprinting. The advantage is the RFLP is considered to be more accurate than the PCR, mainly because the size of the sample used more, use of a fresh DNA sample and no amplification contamination. The limitation is the RFLP, however, require longer time period to complete the analysis and is costly. Next we can see the application of DNA fingerprinting. DNA fingerprinting has broad application in food industry, agriculture. Apart from these, what are the applications? See in the following slide. DNA fingerprinting has various applications in forensic science, paternity and maternity determination, personal identification, diagnosis of inherited disorders, development of cure of inherited disorders, detection of AIDS, and in breeding program. Shows the broad application of DNA profiling in agriculture, forensic science, parentage testing, medical diagnosis, and in food industry. Thank you for watching. See you with another topic in another class.